Thank you, Carlo, for organizing this and giving me a chance to present my postdoc work that I've done in Barbara Tortline's group. And thank you, everyone, for being here and joining me for this talk. So my work has centered a lot on understanding morphodynamic trajectories and shapes in the early organoid patterning. So before I really launch into it, I also want to begin by thanking a lot of people who have helped me in this project. So Barbara and Gray, of course, who have been my postdoc mentors during this journey, but also a big shout out to Fatima, who now gave this wonderful talk right before me. So she has been instrumental in conceptualizing the early phase of this project and also helping a lot with the hashing and different kinds of experiments that she talked about. And another big shout out to Jill, who is my co-author on this study, and he has spearheaded the image analysis side. And I would be giving a glimpse of all the newer analysis that we have developed for light sheet image analysis for the project. So just like a brief overview, although yeah, we had this before. So from a more morphological point of view, brain development begins with the specification of a two-dimensional sheet, so the neural plate. And this neural plate then has like a little and this groove folds over. So the neural plate consisting of the neuroepithelium slash neuroectoderm, this folds over to create like a fold which fuses on the dorsal side. And from this arises the 3D tube-like structure that we call as the neural tube. Now, as all of these morphogenetic events are happening, this shape is getting created. A lot of patterning events are also occurring. So multiple different morphogens are getting secreted from different parts of the growing neural tube or the surrounding embryonic tissue. And these pattern this structure into distinct regional identities. So this would end up making a rostrocaudal and a dorsoventral identity based on the combinatorial expression and the regional identity as we heard before. So now together, morphogenesis and patterning are occurring at the same time as the structure develops. And this results in forming the developing brain as we know, where now the anterior regions would form the big lumen, big forebrain containing part with the identity of the telencephalon and the diencephalon. And as we go more caudally, then the lumen becomes smaller, the tissue structure becomes different, and the identity is more midbrain and so on towards the hindbrain. Now, not only this at the tissue scale, but a lot of huge cell diversity happens during brain development, where, for example, I've put some neuronal diversity here. So also at a cellular level, multiple different cell shapes and cellular identities are getting created as the brain is developing. And as we know, an anomaly in any of these events, from morphogenesis to the cell type specification, can result in several early neurodevelopmental disorders. So a lot of what we know about this process that I just explained is from work done on mouse and other model organisms. But the dynamics of how morphogenesis and patterning are talking to each other during these events has largely remained obscured, simply because I'm talking about events that happen in the first few hours of mouse embryogenesis, where it's really difficult to access live tissue, or 2 to 5 weeks of human fetal development, where again, we don't really know what really happens during these very early patterning stages. So the question that I took up and I really found very interesting is to now figure out how to integrate these how these multi-scale interactions are integrated and what are the dynamics of these processes over time. So fortunately, now in the last decade, human brain organoids have been established as an in vitro model system to model some of these early processes, where from stem cells, we can create these aggregate embryoid bodies and so on. So I am only talking about the unguided human brain organoids in this talk. So I don't really give any patterning morphogens, no guiding cues, but let the system self-organize. So if you do this kind of a protocol, then over several weeks and months, the organoids start showing several lumen, where now if you do a cross section, then at about 2 months, we see that this big lumen, similar to the neural tube that I showed on the previous slide, these luminal regions are created, which also have the right orientation. So very similar to the primary fetal brain at these stages, the organoids have progenitors towards the luminal side, and then there are more neurons on the outer basal side of the organoid. Further, now a huge concerted effort has started integrating a lot of single cell analyses and different kind of atlases that people have produced in the last 10 years to compare these organoids to the primary tissue, where now we have a better understanding of what are the different cell types and what are the different brain regions that are getting created in these protocols and what is really that we are recapitulating. And parallelly, a lot of effort is being made to model early neurodevelopmental or later neurodevelopmental diseases using the organoids. But all of this work has been largely done with snapshot studies. So whether it is single cell RNA-seq or whether it is fixing and staining, we are able to get glimpses into the organoid growth, but we didn't really have any way to understand how this evolves in a dynamic, high spatiotemporal fashion over time. So when I started this work, there was a seminal single-cell transcriptomic time course from Barbara's lab, where they profiled these organoids over 4 months and they with single-cell RNA-seq, and they showed that initially you have an embryonic body largely composed of stem cells. This then undergoes forming a neuroectoderm, neuroepithelium, which over the next 4 months starts making distinct brain regions. So you have telencephalic and non-telencephalic regions, and you also have dorsal and ventral regions showing up, and a lot of neurogenesis happening in this time. However, the big black box now is what really happens in this first month. So how the stem cells, without any guiding cues, are able to self-organize and form all this cellular diversity representing different brain regions. And not only that, how are these lineages organized spatially in the organoid? So what is really regulating? What are the fundamental principles here that pattern these organoid into forming this kind of regional cellular diversity? So to do that, I had a challenge in the beginning where if you were to look at a brain organoid, there are several optical challenges before setting up any live imaging. So by that, I mean, first of all, it's a very slow developing protocol or system where I'm talking about tracking 1 month of organoid growth in a microscope. Then the second challenge is these were really large. So people were making them with 10,000, 20,000 cells, big embryonic bodies that wouldn't even fit in the field of view after 2 or 3 days of growth. Further, neural tissue is notorious for being very optically challenging. It's really hard to, it's not like an epithelial system where you have nice polygonal cells. The pseudostratified neuroepithelium is really hard to image and show cells distinctly. And it's a cell culture system. So we need to grow these organoids in a microscope stably for this long. So I circumvented these challenges by working both on the protocol side, but also testing different microscopes and adapting the chambers, where in the end, what we have set up is a sparse and mosaic labeling method. So we are taking largely unlabeled parent cell line, mixing up with GFP and RFP label, and then growing them in a microscopy chamber where you can image 16 organoids in parallel with 4 subdivisions. So you can do replicates of 4 per condition in if you want to also do like a control and 3 perturbations in the same experiment, or all 16 can be grown in the same conditions. So for this, what I did, now we took up a lot of labeled cell lines and we were mixing these GFP and RFP cell lines. But if you really look at it, all the GFP tag lines are actually targeting different subcellular regions. So we have, for example, histone GFP or an actin GFP. And by looking at it, we know that these are 2 different cells because of where the GFP is localizing. Similarly, you have a membrane RFP, tubulin RFP, or lamin RFP, and this is also very easily obvious to the human eye as being different cells if they are mixed together. So I decided to mix all of these up in the big unlabeled background, so 6 cell lines mixed together, where we now make these multi-mosaically labeled organoids. And the advantage here is that now in a single cross-section, instead of having 1 or 2 readouts and then repeating, we could track multiple subcellular features in the same experiment. So we are able to see tubulin versus the lamin or the histone versus the actin and look at how these dynamics are as the organoid grows. So to give you a picture with this modified protocol, etc., what we have is now a day 4 embryonic body, which we embed in Metrogel, and then we put this in the microscope. We start imaging. This would be a much smaller embryonic body made with 500 cells. And as this grows, this nearly doubles every day in size, till at some point it would stop fitting in the field of view. And here we would start doing tiling to still keep on acquiring our images. And I'm really happy to show you this movie here, which is now this multi-mosaic labeled organoid and 5 labels in the same organoid. So you can see some lamin here, histone, a lot of tubulin. Actin is actually a bit in the back, and this dimmer region is CAX. And this movie is now going on for 1 week of organoid growth, imaged at every 30 minutes. And just by eye, you can appreciate how the organoid has changed over time. So in the beginning, when it is stem cells, these are now loosely lying stem cells with very loose shapes. This then becomes an epithelium, and this epithelium is then growing with the cells elongated and becoming more radiculi-like cells by the end of this movie. So further, we have also extended this for some experiments where we took this NKX 2.1 GFP line that Fatima also talked about, and we imaged this now for 3 weeks straight in the microscope, and you can see that the medial ganglionic eminence regions are coming up, cells are starting to migrate, and a lot of these morphodynamic features can be recaptured in these long movies that we are able to acquire. Another power here with the neural system is that the cells have very distinct shapes, and just based on the shapes, one can start interpreting what kind of a cell it would be, even if it is labeled with a generic label like a tubulin. So what we have with this setup is now 16 parallel positions imaged in one experiment, multiple subcellular labels possible, and then we have gone up to 3 weeks of stable imaging and collecting the data, which ends up being several terabytes of huge data sets. And I'm not going to go into the image analysis side due to lack of time, but happy to discuss this afterwards if somebody is interested. So we had these multi-mosaic organoids where we are mixing GFP and RFP labels, which we can see by eye. So now we wanted to create an image analysis pipeline. And this is the work that has been by Jill, who is a PhD student in the lab, where we set up a lot of pre-processing, which involves registering, denoising, etc. And then we trained a neural network to segment all these cells and then be able to demultiplex these cells. So this is what we were really going after, that from an imaging perspective, it's all GFP and RFP, but once you have the data collected, you will have a follow-up software where you can segment all these cells, and then the network is able to learn how to differentiate between a histone GFP versus an actin GFP and demultiplex these. So this now results in like thousands, like almost like millions of images and cells that we are collecting from all these movies, and we have this over time. So we take the full breadth of our movie and do this for all time points per day, etc., to start getting these features. So as a first application of this light sheet setup that we had developed, we combined it with some lineage tracing done with single cell RNA sequencing. So here we had developed an eye tracer barcoding system where you introduce the barcodes and iPSCs and you also scar it at a certain point, and eventually you sequence it. And based on the sequence, we can find out what kind of lineages are getting created in the organoid. Then we took these bar-coded organoids and we did some Visium spatial transcriptomics where we slice and then we again sequence it, where very interestingly, we found that a lot of clones are regionally located. So all the daughter lineages from the parent seem to be lineage restricted regionally. And if we look at the regions that were coming up in these organoids, they were also getting segregated in a similar fashion. So I asked the questions of how could this be happening? And can I track this with the imaging and see why daughter cells are not really mixing across the organoid? So we took up our light sheet movies, and here you have like a pseudo color for the cells, but the nuclei are in green. And then we took a single lumen and we started tracking all the lineages from a single mother cell over 100 hours of organoid development. So first 4 to 5 days after the neural induction. And very interestingly, we found that all these lineages actually do stay in the same lumen. And now if we compare with the diametrically opposite lumen and also collect two more lineages in the same, then these also remain spatially restricted. So what ends up happening is that all the lineages are in the same region of the organoid, but they are getting segregated as the organoid is becoming bigger, and these are getting kind of spatially separated. So moving on from this, then I wanted to further delve into this initial lumen patterning events that we found. And for this, we did a big HCR screen, which is fluorescent in situ method, where we went to the literature. We collected a list of about 40 to 50 genes, which are known from mouse and other model organisms to be patterning the early brain. And we stained for these genes, and we found that, in fact, it is the first 2 weeks where this initial distinction between telencephalic and non-telencephalic regions starts showing up in the organoid. So this indicated this would be a good time point to go with the live imaging and ask this question of, can we identify what is making these distinct regions as the organoid develops? So as I mentioned, we can image 16 organoids in parallel. And as you all probably know, organoids are notorious for being very heterogeneous and variable. So you can see here, all these are cross sections from one experiment. And in fact, they do look morphologically very different from each other, even when they're all growing in the same chamber. However, if we now start measuring parameters like organoid volume, luminal volume, peak lumen number, then we found that despite the obvious morphological heterogeneity, the growth parameters are actually very consistent. And this indicated to us that there might be an underlying genetic level regulation that is somehow keeping these parameters very consistent. So we did our single cell RNA-seq time courses for these early events, and we found that all the cells in these stages can be divided into an early and a late neurectoderm where there is a gene expression switch. So very early on, when we start imaging, it's largely stem cell markers that are expressed. But later on, a lot of forebrain markers and a lot of ECM related genes start coming up. So this indicated to us that as these organoids are growing, they are starting to secrete a lot of extracellular matrix on top of getting the patterning for the forebrain regions and other regions. So here we wanted to go further into what this extracellular matrix could mean. And what we are doing here is, normally we are embedding our organoids in metrigel, which is this mouse sarcoma derived basement membrane enriched gel, which is actually very uncontrolled. We don't really understand what it does to the organoid. We don't know whether it is needed, and we don't really know what it contains. So normally extracellular matrix would have some proteins that would interact with the tissue. It could have some growth factors being secreted there, etc. So we grew it with matrigel, and this is again a cross section to see how the lumen behave and a 3D rendering to show how the lumen grow over time. Then we grew it completely without any metrics. And the idea here is, if the organoid is secreting some ECM, we wanted to see what is the impact of that on the organoid growth. So we grew it without it, and very interestingly, we found that now there are many, many smaller lumen getting created, and these don't really fuse like they did in case of matrigel. And lastly, to give some sort of a diffusion barrier or a low cage, we provided a low percent agarose to just hold the organoid, but not really interact with it. And this was very interesting because we found that this was kind of in between. So it did create a lot of lumen like no matrix, but it did show that possibly the organoid does interact with the secreted ECM if it is captured and retained. And this ends up then creating these bigger lumen with more fusion events occurring in the organoid. So now to look at the cell level of what it could mean with these fusion events, we developed an alignment index. So we have segmented all the cells in these images. And by alignment, I mean how perpendicular is a cell to the surface of the organoid. So when you expose these organoids to matrigel, within a day, so really fast, all the cells respond to matrigel and they're aligned in a perpendicular fashion to the outside. But if you don't give it, then we found that cells are completely mixed in their alignments. And with agarose, there are regions that emerge which have better alignment. Polarity, we stain for polarity markers like collagens and apical polarity like Adherin2. And you can see that in presence of matrigel, there is a really nice basement membrane that is organized.
Looks like we may have lost her.
Hello?
I think it wasn't you. I think Carlo dropped.
Yeah, it looks like, yeah, I thought it was Akanksha's connection, but it does look like mine dropped. Can you guys hear me now? Apologies. Did that disrupt things?
Where was I? Where did you hear last?
I think ask the others.
Fatima?
Like I didn't stop listening.
For
Like
you, it was okay.
Yeah,
yeah.
so just
Okay,
keep
then
going.
I just continue from here.
Yeah.
Yeah. So what we had done is we had segmented a lot of cells, right? So we generated a list of about 25 to 30 parameters, such as cell volume, cell elongation, curvatures, etc., to now divide these cells in clusters based on their morphometric parameters. This means that each cluster contains cells which are similarly shaped. And now we try to bring some single cell analysis tools to this live imaging. And if you make a UMAP out of it and a Paga plot, you see that over time, so from like this, how this plot changes, the darker colors are later time points and the lighter colors are the earlier time points. So over time, the organoid contains cells which are elongated their axis. So this means that the yellow clusters are the earlier clusters in the organoid, which are mostly composed of smaller, more rounded cells. And over time, this is elongating and becoming these more cyan, dark cyan clusters, which have more radial glia-like cells or some other shapes coming up. So very interestingly, when we now look at this morphotype distribution between our different ECM conditions, we find that in presence of an external matrigel, the morphotypes very gradually shift and you lose all the earlier rounded clusters to make all the cells more elongated and radial glia-like. But in the absence, a lot of mixed clusters exist and this gradual transition is not really happening. And agarose was again showing in-between behavior with a few more elongated clusters compared to the no matrix. So then we wanted to know that, okay, the tissue architecture is different, the cell morphotypes are different. What does it mean to the patterning of this tissue? For this, we took up our 3 conditions after the imaging was done. And now we did a single cell RNA-seq experiment at this point to compare them. And all you need to pay attention is to these 3 clusters. So when you provide matrigel and you have this matrigel guided path of organoid development, we find that a lot of telencephalic progenitors are largely coming up in the organoid. However, if you give it without any matrix or you give the agarose, we find that a lot of neural crest starts coming up and a lot of diencephalic progenitors are coming up with agarose showing this entire distribution and an in-between situation. So when we do a differential expression analysis between these 2 conditions, we found that one of the top DE genes was Wnt-less. And this was really high in the no matrix organoids. Now, Wnt-less is the Wnt ligand secretion mediator, and Wnts are required for caudalizing of the developing brain and caudalizing of the developing embryo. So this made us ask the question that could it be Wnt-less driven behavior that creates more diencephalic and neural crest in the organoid? For this, the first thing we did, we combined the single cell RNA-seq with the iterative immunohistochemistry experiment, where we take a glass plate and we put a lot of 2D tissue sections from different conditions over time. And then we do several cycles of antibody staining, so wash and stain cycles, where in total we had about 70 antibodies in this experiment. And then we do the image analysis where we segment all the cells and we divide it into a subcellular fraction, cytoplasmic, nuclear, or extracellular matrix fraction. And this is just an example to show you that we collected now this list of 50 plus genes, which would either be a patterning marker or a polarity marker or an extracellular matrix component. And when we do this, we find that indeed, in the beginning, the organoids in both conditions are largely very early neurectoderm, unpatterned. Now, matrigel still says unpatterned by 2 weeks, but by 3 weeks, it starts showing this telencephalic patterning, so these cyan colors. But very interestingly, it's the central regions that start showing non-telencephalic regions come up. Whereas if you grow it, without any matrigel, very early on, the organoid starts differentiating into non-telencephalic regions. So all these blues correspond to non-telencephalic clusters. And in fact, this orange is neural crest. So a lot of neural crest also starts showing up in the organoid. And then by 3 weeks, we see a lot of PNS neurons, a lot of non-telencephalic neurons, and largely diencephalic patterning emerging in the organoids. This is just something very new, so I wanted to give a snapshot here. Now we could do the same thing with the extracellular matrix gene. So we have about 7 ECM proteins like collagens, laminins, etc. stained. And then we could take up this ECM fraction and we found that there is also a very different correlation between ECM composition and the different telencephalic versus diencephalic progenitors that are coming up. So last thing we wanted to do is we wanted to now find this link of how extracellular matrix could be causing this patterning different between the 2 organoid conditions. And what is the role of Wnt-less in this process? So one of the genes which is very well studied as a mechanical transducer, but also an ECM sensor is YAP1. We stained for YAP1 and we found that YAP1 is very high in the no matrix organoids. And it is known from mouse cardiac progenitors that YAP1 upregulates Wnt-less expression. So we also did a cut and tag experiment to now find in our neural system that YAP1 indeed binds to the Wnt-less promoter. And then we provided a YAP1 activator to see what happens to the organoid. So normally the organoid, which is the control organoid, would have these big lumen as I've shown before. But if you activate YAP by providing a YAP activator, the organoids start completely rounding up. So all these lumen collapse. And in fact, the morphology starts looking very similar to the no matrix morphology. And if we compare the patterning differences between these 2 conditions, we found that Wnt-less is very high when YAP1 is given to the organoid. And again, now these organoids start having a lot of non-telencephalic regions. So YAP1 is upregulating Wnt-less, and Wnt-less likely is impacting the non-telencephalic region emergence. So to fully seal this point, we did an experiment where we now created a new knockout line for Wnt-less. And we grew organoids from control and knockout line, and we gave them different treatments. So we grew them with matrigel, without matrigel, with a DMSO control, activating Wnt by a Kir pulse, and activating YAP by adding the Pi60 activator. And we did a hashing experiment as Fatima showed in the previous talk. So when we did this, we found a huge cellular diversity is getting created in all of these organoids combined together, where they are forming telencephalic regions marked by FOXG1, but also these Wnt-less regions, choroid plexus, etc. So all we need to pay attention is to this 2 conditions where we have now either activated Wnt by giving KIR, or we have activated YAP by giving pi60. And as you can see in the control organoids, upon Wnt activation, a lot of diencephalic retinal or choroid plexus regions are coming up, and this does not happen in the knockout. So Wintless is indeed required for these diencephalic regions to come up. But also very interestingly, if you give a YAP activation, neural crest is up, non-telencephalic mid-hindbrain regions are up, and as well as diencephalon is up. Again, in the knockout, none of these diencephalic regions are increasing, but some of these mid-hindbrain regions were still higher, indicating that YAP could possibly have an independent role here, which is independent of Wintless in creating the posterior regions of the developing brain. So to summarize, what we have done in our project is we have set up a new light sheet imaging-based morphodynamic profiling and analysis method where we can track tissue scale parameters like lumen, volumes, and tissue volumes, but we can also track cellular morphology, cellular trajectories. We have combined this with single-cell RNA sequencing to profile what kind of gene expression changes are occurring. And what we have also done is the spatial antibody parameters to start combining the molecular changes in the organoid. And with this, what we find is that upon exposure to an external extracellular matrix source, stem cells organize into an aligned and polarized neuroectoderm, forming this neuroepithelium that ends up forming big lumen and patterning to telencephalic regions. If we don't give this, and if the organoid is grown without any external source, it upregulates its YAP1 expression, possibly due to some mechanical changes or some ECM changes within the organoid. This YAP1 is also upregulating Wintless expression, and now it starts patterning to different regions of the developing neural tube. So now a lot of neural crest, but also a lot of diencephalon and mid-hindbrain regions start coming up in the organoid. So this is just a little bit of teaser of where these kind of projects go next. So now that we have all these morphotypes segmented, we are tracking them with some AI-based automated tools. And with these trajectories, we can track cell divisions. So this is just blinking cells where each cell is a newly divided cell. Or here in the bottom movie, it's color-coded, where every subsequent lineage is getting a newer color. So blues are the latest cells, and then red was the earliest. Lastly, we are assigning these cells to different lumen to see which kind of regions they belong to and what kind of shapes they have. And what we be doing with this is to start compiling these lineage trees. So starting with our yellow initial morphotypes, how do you end up getting these different diddle glia and different progenitor populations, and what are the molecular states in these populations? So this is more the future direction aspect of this project. So with that, I would like to stop and thank all the people again, also especially Viventis Microscopy, who I have worked a lot with them to optimize the microscope, the different BSSC facilities at the ETH, the entire team that worked with me, Jill for all the image analysis side, as I said, and yeah, my supervisors for mentoring these projects. Thank you.
Thank you, Akanksha. Question? Really incredible imaging.
Arlo, can I get the ball started?
Yes, please.
I want to just start by saying to both of you, it's just totally spectacular way of spending an hour or two listening to you talking about these cells. But if I can just ask a question, which is very sort of, you get sort of blown away by all the beauty of it, right? It's like reading Ramoni Cajal. You see the full glory of the spatial organization, is how he saw it initially, of the brain. But about a third of the way into your talk, you had a very simple slide where you showed that there was two stages of neural differentiation, very early stages of neural differentiation. And I wanted to ask you if you think that slide and that simplicity captures all of the lumen diversity that you ended up showing us. Is it all captured within those developmental constraints, which you measured? You showed gene expression.
Yeah. So actually, I think what I have come to realize is that there are possibly two different things happening here, which maybe are independent. The morphogenesis seems to be an independent behavior, which is creating these lumen, creating these elongated cell types. And this is more the self-organized aspect, creating very simple cellular population of early or late neuroectoderm. However, the morphogen guidance that happens on top, which in my case was the self-organized morphogen production, Wintless increase, or in Fatima's case was provided externally, this possibly overrides the inherent simplicity and then starts patterning it to different regions. That's what I think at the moment. Did I answer you or?
Yeah,
You're-
I'll just respond quickly to that. So it's very hard to really communicate clearly about such, all beautiful properties of these emerging epithelia. But when you talk about the choroid plexus in that piece at the end,
Yeah.
and of course, it's probably worth everybody remembering that the choroid plexus is the first part of the prosencephalon that actually becomes specified. And actually, if you block Wnt signaling at that stage, you get holoprosencephaly. So you get this huge expansion of this very simple cell. So it seems to me that what we're looking at here is a cascade of events which have really highly conserved properties.
Yeah.
But they're all playing out in these different regions to generate really quite specific sets of neurons with their own different functions. And everything seems to point to this being the precursor cell that's actually doing it, right, making these differences. And I think we should talk more about why that is so, right? Why is it designed this way? TBR1, for example, Fatima, in your beautiful image of those things you called regulons, TBR1 is sitting there. That's a high-level autism risk gene. It's up in that top left-hand corner, which is sort of forebrain. And I could talk more about it, but it seems to me worth our while realizing that we're all kind of really trying to say quite specific things about this system and how it works. And when it doesn't work properly, well, there are consequences, apparently. Yeah, I hope that's useful. I think the Chris Walsh Wnt1 story is in the mouse. It's like, I mean, it's embarrassing. It's like 20 years ago. Clear as day, though. And then he worked out how the choroid signals to the subsequently generated ventricular radial cells very specifically. And then you showed beautifully they stay together. They have to stay together because they must play together. And in your lineages, I'll say one more thing and then I'll shut up. In your lineages, do a lot of lineages just simply disappear?
Yeah. In the live imaging, not. In the live imaging, the tree stays, but we have not gone that long in the live imaging. We have done about 4 or 5 days of live lineage tracking in the first part that I showed where they did not mix. Beyond that, now we are trying to go for more like 2 or 3 weeks when this patterning events have really come up. Because what we are missing in the current story is this transition from the early morphogenesis to the appearance of the first patterning progenitors.
Thank you.
And
Thank you so much, Jed. Yeah, go for it.
just to add on to that, another thing which we are missing, which I think is just nice to bring up, is this kind of regional reporters to mark some of these regions or some of these cell types that you're mentioning, and then really track it in live to see which now some of them, if they really disappear or do they actually end up moving around. Yeah. So we need more reporter lines for that. Yeah.
Thank you so much, guys. I've really enjoyed not only your talks today, but this whole series of talks we've had recently on morphogens. It's a hugely complex problem, and as many other studies have showed, is not only interesting to understand how the brain is regionalized, but also, as Ron mentioned, it's increasingly obvious that as these very early processes go awry, it's at the root of a lot of neuropsychiatric goings on. So super interesting to understand them. Thanks again, Akanksha and Fatima. We'll see you next time. Oh, we have one more question if you'd like to jump on it. Here we go. Can you see the chat or would you like me to read it?
Yeah, I saw it. No, I think 1 or 2 more channels, such as in the far right, can be added. And more recently, I have been testing this if I can do 4 in parallel or 5. But I do think for the very long term, like 3 weeks, this would increase photobleaching. And I rather want to stick to this combinatorial mosaic approach then, where different cells could be demultiplexed based on nuclear or cytoplasmic expression and then be tagged to some sort of a regional reporter. Because I think at the moment we have done sequencing after 2 weeks of imaging, and the organoids did not show any too much dying or any photobleaching, phototoxicity. But at 3 weeks, 4 weeks, the kind of timelines that I'm trying to push for, I don't think it's a good idea to add too many laser channels.
There's also one more that we missed in the first round. I see, Fatima, you responded to him. Cool.
Yeah, sorry, I didn't know if there was time or not.
Yeah, I'm happy for you to address that right now. People can take off or stay as they please.
Fatima, go ahead.
Sorry, I missed that. Yeah, I don't know if Jin Chung is, yeah. So yeah, basically we include GSX in the analysis, like as one of the other markers of ganglionic eminences. Like we take into account all the possible genes, and I would say both the MR and the organoids recapitulate quite well ganglionic eminences, and if anything, we're a bit biased towards the ventral sides, and we have less of cortex. Like we don't know how far we go above the pallial subpallial boundary, but I think ganglionic eminences are perfectly there.